So I'm sharing the screen, and this is just on the aspects of hearing. In terms of hearing, you have sound. Sound travels in waves. These waves have a certain frequency, and they have a certain intensity in terms of very loud sounds, very deep sounds. All of that can change. You can have like a deep, low, or deep, loud. It all varies. So you have these sounds travel in air in ways. You can actually just think of air as being a non-fluid liquid is really what we want to think of water. You can look at water and you can throw a rock in the water. You can see the ripples. Depending on how strong you throw the rock in, that is going to alter the amount of ripples in the water and how fast they're moving. If you throw the stone in slow, it's going to cause little slow ripples to occur. If you throw it in real fast, it's going to make the ripples really close spaced together. So we can affect the amplitude and the frequency of these airwaves. And that's what sound is. It's affecting the amplitude and the frequency. And this fluid wave is going to come in, and it's going to be collected by the auricle of your ear. And that's going to help to channel it into the external ear canal, the external acoustic meatus. And at that point there, it's going to hit the tympanic membrane. Now, we're going to convert that fluid wave into a physical wave. If you put your hand on your desk and put your hand on your desk and knock on the top of it, you can feel the transmission of that wave coming through the desk. Whether you're doing a light tapping, I'm doing a light tapping on my desk right now, or a hard thumping, you can feel that transfer through the desk. It's no longer a sound. Well, you can still hear it. But as you're conducting it through a solid, you have converted it into a physical phenomenon. And that's what the eardrum does, is it converts the sound wave, the fluid wave, into a solid physical wave. And that eardrum is connected to the malleus, and the malleus is going to vibrate back and forth onto the malleus, the incus. And the incus is going to transfer that vibration to the stapes, the stapedius, and that's going to transfer it to the oval window. And then inside the oval window is just a membrane. As that membrane is vibrating back and forth, it's going to transfer the signal back into a fluid. But in this case here, it is a liquid fluid that we're transferring it into. And then that fluid is going to go through the cochlea. And in the cochlea, there are going to be different regions. Now, we have like these, the tectoral membrane and the basilar membrane. Now, that's the structure between the two canals, large canals, the basilar canal and the tympanic canal. There are different sections along it, and they're designed with different thicknesses. If that's vibration in the fluid can cause that particular structure in one part of the ear to vibrate, then the sound will be transferred into that little segment. And it will cause another membrane to vibrate, the tectoral membrane. And as the tectoral membrane is vibrating, there are hair cells. So the tectoral membrane is vibrating, a hair cell will be vibrated. That hair cell, when it's vibrated, is going to create an action potential. That action potential is going to go to your cochlear nerve, and cochlear nerve to your head, and you're going to interpret that sound. And then that sound wave is going to go out back into the canal and come back out. Energy comes in, it has to go back out. And that's what the round window is for. So we transfer that energy from like one fluid to another fluid back to the original fluid, and then it gets the round window, and in the round window, the membrane vibrates, and it returns the sound back into the middle ear where you have your Eustachian tube, the auditory canal. And that's just filled up with air. And that will allow that pressure to dissipate by going out into the just above the oronasal pharynx is where that sound wave comes into, sort of like just inside, just past your nasal cavity. That sound wave goes in, because otherwise, if that Eustachian tube is blocked up, that means that sound wave is going to be hitting the eardrum again. You have like the sound coming in the eardrum this way, and if the sound's trying to go back out the same way, that's going to cause interference to occur. Have you ever gone to a speaker and put your finger on the speaker? Like the speaker is vibrating back and forth. Have you ever gone and touched the speaker and the sound sounds muffled? Well, that's because you are interfering with the speaker's ability to vibrate properly, and the same things happen with these sound waves. Is the sounds trying to come in the eardrum and also go out at the same time, and you have like these two opposing sounds, and they're trying to alter or cancel each other out in some aspect.
Many of us take for granted a very extraordinary organ, our ears. To understand the ear, we need to understand what sound is. The speakers you are listening to right now are vibrating, flexing in and out, causing a wave of pressure through the air. The frequency of these waves, or the speed at which the sound creating surface moves back and forth, affects the pitch of the sound. The level of air pressure in each wave is directly related to how loud the sound is. The outer part of our ear catches these waves. It faces forward and has a specially designed structure of curves, helping us to determine the direction of the sound and also emphasize the frequencies used in human speech. Now that the sound waves are caught, they travel through the ear canal and strike against our eardrum, a thin membrane about 10 millimeters wide. Now that we receive the sound, the middle ear transfers this energy. The smallest bones in your body, the malleus, incus, and stapes, start in motion. The malleus is attached to the eardrum, and as the sound travels along, the force is amplified by leverage until it arrives at the stapes, which acts like a reversed piston, creating waves in the fluid of the inner ear. The most significant increase in pressure is caused by pneumatic
Look what's happening over here. Pressure's coming in on this side over here. It's going to be transferred this point over here and go down the channel at that side over here. The returning channel is going to be on this side over here, and that's where you have the round window. And watch, every time this one pushes in, this one's pushing out, because it's a very fast process. The energy comes in, gets around, comes to a certain point, crosses over, comes back around, and comes out. But this over here is going to allow that pressure that's in the fluid inside of the inner of the cochlea to exit. So the energy comes in over here, pressurizes the fluid, and then the fluid is going to release this pressure at that point there into the space over here. A Eustachian tube is down over here, and that's going to allow the energy to escape rather than that energy just coming back over here and hitting the eardrum again.
Amplification. The face of the stapes has a surface area of about 3.2 square millimeters, while the eardrum has a surface area of 55 square millimeters. Using this, along with leverage to the malleus and incus, the final pressure is 22 times greater than when the sound first arrived. Now we come to the most complicated part of the hearing, the cochlea. In reality, it is coiled up, but it is easier to understand straightened out. There are actually 3 chambers inside, but let's take a look at the central part. The stapes is causing pressure waves to travel through the structure. Along the inside wall is about 20 to 30,000 reed-like fibers. As the waves move along, they encounter fibers with the correct resonant frequency and energy.
So different sections of this vary in thickness. And only certain wavelengths are going to cause this section to vibrate. A different frequency is going to cause this one to vibrate. A different frequency will cause that one to vibrate. So if the energy can cause it to vibrate, the energy is going to come out and into this location over here.
He is released. These fibers aren't actually what give us the signal that we heard something. There's a special structure next to these fibers containing hair cells. When the fibers resonate, they cause the hair cells to move, which then sends an electrical impulse to the cochlear nerve and onto the brain. Certain pitches of sound will resonate in specific locations, and louder sounds will cause more hair cells to move. Our brain interprets all this raw data, making it possible to enjoy things like music or an engaging conversation. Just to think that all of this is happening in your head right now at full speed. This is just one of the amazing systems found in the human body that go far beyond our
Yeah, I wish they would have talked about how the sound is transferred from, let's say, this section over here. Energy will end up over here. Once the energy is over here for a specific frequency, then this area, this over here is pressurized. We have to get that pressure out of it so that we can respond to, it can respond to new sounds. So we have to transfer the sound out, the energy out, and the energy gets into this one over here. Which is
human understanding.
sort of shown in the next video a little bit.
People with ADHD save 10 hours studying with this Chrome extension. But no, by all means.
As sound enters the ear, it passes through the external auditory canal, where it meets the tympanic membrane. The tympanic membrane then vibrates in response to the sound. Sounds of a lower pitch or frequency produce a slower rate of vibration, and sounds of lower volume or amplitude produce a less dramatic vibration. Higher frequency sounds produce faster vibrations. The tympanic membrane is cone-shaped and articulates with a chain of 3 bones called the auditory ossicles. They consist of the malleus, the incus, and the stapes. The movements of the tympanic membrane vibrate the ossicles, passing on the information of frequency and amplitude. The 3 bones pivot together on an axis shown here in red. The pivotal axis is due to a series of ligaments which hold the bones in place within the middle ear cavity. The anterior malleal ligament and the posterior incutal ligament are of particular importance for the pivotal axis. 2 structures which normally obscure this view of the middle ear have been removed. They are the chordae tympani nerve and the tendon of the tensor tympani muscle. Through the ossicles, the vibrations of the tympanic membrane are transferred to the footplate of the stapes. The stapes moves with a piston-like action, which sends vibrations into a structure called the bony labyrinth. The labyrinth is filled with a fluid called perilymph. If it were a completely closed and inflexible system, the movement of the stapes would be unable to displace the perilymph, and therefore unable to send vibrations into the bony structure. Due to the flexibility of a membrane called the round window, the stapes movement can displace the perilymph, allowing vibrations to enter the labyrinth. The corridor leading to the round window is found within the spiral portion of the bony labyrinth known as the cochlea. Vibrations produced by the stapes are drawn into the spiral system and return to meet the round window. The portion of the spiral passage in which vibrations ascend to the apex of the cochlea is called the scala vestibuli. The descending portion of the passage is called the scala tympani. A third structure, called the cochlear duct, is situated between the scala vestibuli and the scala tympani. The cochlear duct is filled with a fluid called endolymph, and when viewed in cross-section, the membranes separating the 2 fluid-filled systems are visible. They are Reissner's membrane and the basilar membrane. The membranes are flexible and move in response to the vibrations traveling up the scala vestibuli. The movements of the membranes then send vibrations back down to the scala tympani.
What fluid is over here? Seeing if we're paying attention. The fluid over here is called the what? Is anybody there?
The Wayland Symphony?
No, the fluid. This is the scala vestibula. This is the scala tympani down over here.
Endolymph?
No, it's the perilymph. This is the endolymph in here.
A specialized structure called the organ of Corti is situated on the basilar membrane. As the basilar membrane vibrates, the organ of Corti is stimulated, which sends nerve impulses to the brain via the cochlear nerve. The actual nerve impulses are generated by specialized cells within the organ of Corti called hair cells. The hair cells are closely covered by a structure called the tectorial membrane. As the basilar membrane vibrates, the tiny clusters of hairs are bent against the tectorial membrane, triggering the hair cells to fire. The entire basilar membrane does not vibrate simultaneously. Instead, specific areas along the basilar membrane move variably in response to different frequencies of sound. Lower frequencies vibrate the basilar membrane closer to the apex of the cochlea, whereas higher frequencies produce vibrations closer to the base. This arrangement is known as tonotopic organization. Together, this sequence of events is responsible for our acoustic perception of the world around us.
All right, so that gives you a fairly good idea of what is going on, correct? In terms of the way the ear is functioning. So now it's just a matter of just going over the salient aspects of it. So again, you've already talked about the structure of it. We've seen how the eardrum is vibrating. The sound waves come in, the eardrum is going to vibrate with a certain intensity, a certain frequency for every sound wave that comes in. That sound will be transferred by the ossicles to the oval window, and the oval window will pressurize the perilymph within the Moni labyrinth. And then within that, so the auricle, we've got the structure there that surrounds, protects the external acoustic meatus, auditory canal. That provides directional sensitivity. It's where you hear a sound and it's not quite clear. You might move your head to see if it's a little bit louder. And as that sound gets louder and louder, you know that the ear is facing the right direction. That also gives you a good visual cue of where it is, because now you hear the sound is like loudest back over here, and you can now turn your head and actually look to see what's making that sound. So it allows you to home in on sounds with your eyes, especially since you have two ears, just like you're seeing with binocular vision, you're also hearing in stereo. So you hear a sound out in front of you somewhere, and you know, well, it's not coming directly in front of you. It sounds like a little bit louder to this side over here, or a little louder to that side over there. That allows you to divert your eyes in that direction, and then move your head in such a direction that the sound gets the loudest. And now you can actually look in that particular location and see what is there, hopefully Unless it's a bird, then they're hiding in branches. You can never see them. He's an instructor. It was a field course, like 30 days out in the desert. He's always saying, "Okay, let's go bird watching." Like at 5:30 in the morning, and go watch, like look for birds. And he says, "Look at that bird over there." I go, okay, got my binoculars. All I see is a tree. Where's the darn bird? Tympanic membrane, the eardrum. That's at the end of the external acoustic meatus, separates the external ear from the middle ear. They're also present in the external ear is the ceruminous gland, and this is a gland that secretes a waxy material. If you remember a long time ago, we talked about some of the secretions that glands do and the way they do it. In the case of this one here, the cell keeps growing, but as it's growing, it starts secreting wax into vesicles. And then a segment of that cell is going to start to fragment. And those vesicles break open, and the wax is now exposed to the passageway. The benefit of that wax is that it is going to trap any foreign objects or insects or bacteria that get into this ear passageway and prevent them from getting up to the tympanic membrane, which is going to protect the tympanic membrane. Now, this wax does not stay waxy. It dries out at a certain rate. And it's actually designed to dry out, and like you turn your head to the side and it falls out of your ear. That's the way it normally should work. What that does is any type of dust that's getting in there, any bacteria that's in there, will just fall out with the wax as it dries up and falls out of your ear. And then also the wax has properties to it that is going to slow the growth of microorganisms, which is going to inhibit their ability to proliferate and then either get into the cell structure within the ear or get up to the eardrum and cause damage to the eardrum. But then also when the wax falls out, the bacteria go with it. So that's a benefit in terms of part of your immune system right there. To protect the internal environment. Remember, once you get inside the inner ear, into the middle ear, and then the inner ear, you are essentially just there's the only thing separating the brain from the bacteria, the pathogen, is the nerve itself. And they can just get onto that nerve, move along the nerve, and get into the brain tissue. So it's really just the tympanic membrane that's protecting your brain from being invaded by pathogens, as well as the immune cells that hang out in the acoustic meatus, the internal acoustic meatus, which is not the internal acoustic meatus, the Eustachian tube. The internal auditory canal, the Eustachian tube, that's actually open up to the throat, but there are many immune cells that are on the mucous membrane of that structure there. Middle ear is a air-filled chamber, and that is going to communicate with the nasopharynx just above the oropharynx via the auditory tube or the Eustachian tube, and that is going to allow the pressure that comes in. The eardrum vibrates, it pressurizes the air-filled chamber over here. That pressure is transferred to the oval window, to the paralymph, and then back to the round window via the paralymph, and we have to get rid of that pressure somehow, and that pressure is going to go out of the auditory tube and prevent it from interfering with the sound waves that are trying to come in on the tympanic membrane on the eardrum. Then there's the auditory ossicles. They are stabilized in their positions by the different stabilizing ligaments, as well as the tensor tympani. And what this one does, it always has a bit of contraction to it. And what that contraction does is it pulls back on the malleus and keeps the eardrum taut so that it's always responsive. Have you ever had a speaker that has part of the speaker edge broken? And when it's making noise, it just sounds like a lot of distortion in the speaker because part of the speaker is broken. It's not held tightly in place, so it just vibrates a little bit too much and makes odd noises. So what this does is it's always keeping the eardrum in a tight position, is what the tensor tympani does. And then the other ligaments help to stabilize it, but they do allow a rocking motion of the malleus to occur. And the malleus, every time it's moving, will impact the incus, and the incus is also suspended by stabilizing ligaments. And it is also connected to a muscle, which is going to help to keep it pulled away from the oval window via the stapedius, the stapes over there. That this keeps it tight over here, so that it does have a bit of give, but it's always pulled away. But then it can vibrate, and when this over here vibrates, pushes inwards, and is going to push on the oval window and transfer the vibration to the perilymph inside the bony labyrinth. So these ligaments, these muscles are important in terms of the stapedius and the tensor tympani in terms of how they always keep this structure here taut, and this one here taut and pulled away, but allows it to enough give to impact the oval window. Then also there's a round window where the energy comes out, but rather than energy going this way here, energy is going to go that way down there through the auditory tube. So when the sound waves vibrate tympanic membrane, the auditory vesicles will vibrate, and they will transfer the sound waves to the inner ear via the oval window. And then I've already talked about the tensor tympani and the stapedius muscles. In terms of the bony labyrinth, that is divided up into the vestibule. That's where the round windows, oval windows are. That's where the cochlea and the semicircular canals will merge together. We'll talk about this structure here in a while, but right now we're just going to focus on this over here, to where the oval window is at this point up over here. You don't really see the oval window, but the pressure wave is going to be transferred to this passageway over here, come around, go around, and then eventually the pressure wave will come back out this way over here. But remember that pressure wave, depending on the frequency of it, like high-pitched sounds will travel from this side here to this side over here very early. If it's a very low frequency, it is going to, like the deep bass sounds, they're going to transfer over at this point up over here. So the vestibule, that includes the saccule and the urticle. There are receptors in here that will detect gravity and linear acceleration. So as you're moving forward or backwards or side to side, the fluid in the vestibule is moving back and forth in various directions, and there are ossicles that otoliths that are in this fluid, and as these otoliths are contacting hair cells in that, those hair cells are being displaced. If those hair cells are being displaced very slowly, that means that you're moving in a certain direction in a slow manner. But if the hair cells are being triggered very quickly, bang, 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 that means you're moving very fast. So you can detect the speed that you're moving at in any direction. You can also, via the semicircular canals, also be advised into how your body is moving, whether your body might be going this direction or that direction, or if it's going this direction or that direction, or if it's going in that direction like that. Because also in the semicircular canals, there are the otoliths that are present. And then the cochlear duct, that is receiving the pressure waves that are being transferred into the perilymph. And within that, there are the organa corti, where you have the hair cells, and depending where those hair cells are, if that segment of the organa corti is vibrating, that will cause the increased movement of the hair cells. And depending on the intensity of them, that's going to affect how many hair cells will be affected, and that will indicate loudness along with the frequency in that particular region. Equilibrium, that's a state of physical balance, sensations by the receptors of the vestibular complex and the semicircular canal. So that's, you get your equilibrium from the vestibular complex and the semicircular canals, as I just talked about, in terms of the rate that you're moving at and the position your head is going in. And within these structures are the hair cells. These are the sensory receptors, and they provide information about the direction and the strength of the mechanical stimuli. In terms of the hair cells, they occur in a complex, vestibular complex, and there are 80 to 100 stereocilia per complex, and they sort of look like very long microvilli. It's just a cell with cells with long microvilli, a cell with long microvilli that are present on it, and a single large kinocilium present on it. So this is just the general structure of a hair cell over here, hair cell over here, and each of these hair cells is connected to its own individual nerve, which is going to lead to, so we're talking about this location over here, this is going to lead to the vestibular nerve. The nerves that are over here will go specifically to the cochlear nerve, but anything over here and over here will go to the vestibular nerve, which will lead to the vestibular cochlear nerve at that point up over here. But in each of these canals, there is going to be a vestibule, and within this vestibule will be the fluid. And then with this fluid, there will be stones, and these stones will be impacting or contacting these hair cells. Depending on where the stones are contacting, it will be an indication of position of your head, as well as how fast the fluid is moving within this complex over here. So again, there are the otoliths. These are the ear stones, and they are just structures of calcium carbonate crystals. They are floating in the fluid, which is the lymph, and that lymph is not it's more like a gelatinous mass, but it still allows the stones to move back and forth through it, and it's going to be detecting your position. Also, it's detecting if you're falling. You actually get that sensation occurring also. So here are the otoliths over here. So every time you're moving your ears to one position, you'll start to have an accumulation of otoliths. These otoliths will be moving back and forth, and these hair cells will be picking up the information. And depending on how many triggered, that will give an indication of the position of your head or how fast they are moving in any general direction. So if your head normal upright position, you have gravity pushing down on the otolymph, and that's going to stimulate these hair cells a little bit. But if you tilt your head backwards, the otoliths are going to move, and you are going to have a greater amount of trigger. This one here is saying, I'm being affected quite a bit, and this one here is saying, I'm being affected also. And then other hair cells are giving other information about the degree of movement that's occurring. And that causes distortion to these cells. They are going to send a signal out at that point there. Okay, let's see. So pathways for equilibrium sensations. All of this, monitoring these hair cells, is going to go to the vestibular complex, which is part of the vestibular cochlear nerve, which is cranial nerve number 8. And in terms of the information here, this is going to integrate the information for about balance, equilibrium from both sides of the head, and that's going to relay that information to the vestibular complex, to the cerebellum. And the cerebellum is going to look at that information and say, do I need to correct any muscles to keep the body from falling forward or keep the body from falling backwards? Or you're starting to tilt to the right too much. Do I need to adjust any muscles to maintain your upright position? So all that information goes to cerebellum. Cerebellum is quickly responding to the information and helping to maintain your body position so you don't accidentally fall. And then we'll send that information to your cerebral cortex and make you aware of what's going on. In terms of hearing, sound waves are converted from a fluid wave in the air to a mechanical wave by vibrating the tympanic membrane, vibrating the auditory ossicles, and then converting it back into a fluid wave via the perilymph and the endolymph in the cochlea. And then there are the hair cells, which are in the organocorti, which is going to be each group of organocorti will be picking up a particular frequency as well as the amplitude or the loudness of that. If the amplitude is very low, a very low amplitude, can you hear me? You can hear that amplitude. I'm talking very softly, but that's amplitude, but just a few of those hair cells responding. Or if I'm talking very loud or someone's yelling in your ear, a lot of them are vibrating. And then this information is going to be sent by the cochlear nerve via cranial, the vestibular cochlear nerve, cranial nerve 8 to the auditory cortex of the brain. So in terms of pressure waves, that's a sine wave, sort of like consistent region where the air molecules are crowded together. In terms of pressure waves, they're going to have a distinct pitch. That is your response to the actual frequency, whether it's a very high pitch sound or a very low pitch sound. High frequency or low frequency. Also the amplitude, that is the height of the sound. Whether it's like very, very loud sound or very, very soft sounds. And then also the intensity is measured, which is a combination of pitch and amplitude combined together, and that's reported in decibels. So information comes in on the oval window and circulates through the canals, whether it be the information comes in on the, or actually comes in, the information, the sound waves come in on the scala vestibuli and the sound goes out on the scala tympani. This is the perilymph. Which is a continuous fluid, but some of that energy can cross over into the endolymph. The endolymph is in the middle duct, which is the cochlear duct, is the middle duct. So remember the three ducts present. The duct where the energy comes in is the scala vestibuli. It crosses over, depending on the frequency, into the cochlear duct and then goes back out into the outer passageway, which is the scala tympani, and now the energy is going to be dissipated out the round window. And then the organocorti, if this membrane over here is responsive to it, then that is going to be transferred to the perilymph, and the perilymph will cause this tectoral membrane over here to vibrate, and that vibrate will impact the hair cells over here. And depending on how loud the sound is, if it's a very soft sound, then they're going to be vibrated mildly, but you still get that frequency. But if the frequency is very very loud, very strong, then it's going to displace more of these hair cells, and the frequency is going to sound very, very loud to you or very, very soft to you, depending on the degree of movement of this membrane in that one particular location. So remember, coming in on the scala vestibuli, going out on the scala tympani. The scala tympani is the out route. The vestibuli is the in route. And then the frequency will cross over in different locations at the cochlear duct. Low frequency sounds towards the end, high frequency sounds at the beginning, more towards the beginning. So 6 basic steps of hearing. Sound waves arise to tympanic membrane. Tympanic membrane is going to displace the auditory ossicles. The ossicles vibrate the oval window. That is going to create a pressure wave in the perilymph of the scala vestibuli, and that pressure wave in different locations will distort the basilar membrane, and that will allow it to cross over into the cochlear duct where you have the endolymph, and that will vibrate the tectoral membrane, which will cause sound, and then that energy will release back into the tympanic, the scala tympani, so that way the energy can transfer back out to the round window. So as the basilar membrane is vibrating, that causes the hair cells to vibrate against the pectoral membrane, and that is going to allow you to perceive the different frequencies of sound. So this is a model that's this is a model they actually use in the classroom over here. It shows like here is the scale of vegetability. This is where the energy comes in. This is the paralimp here, paralimp here. This is a continuous network from scala vegetability to scala tympani, but in different locations, some of the energy can vibrate this membrane over here. If it vibrates that membrane there, the energy is transferred to the endolymph over here, and the vibration will affect the tectoral membrane at that point there, and then that energy is going to be released back out into this zone over here. Scala tympani. Why do I have that backwards? Scala vestibuli, energies coming in on the vestibuli. These are backwards. This should be the scala vestibuli. The comes to that should be the other way around. I didn't want to move that. Hold on, let me just fix this over here. I got on my way. There. I don't know how that got altered like that. There we go. Okay. And then the cochlear is the middle one at that point over there. Scala, why is that up over there? That should be down over there. So in terms of the energy coming into the oval window, the high pitch are going to be heard early on, and the low frequencies, the bass sounds, are going to be heard much later at that point over there. And it's just looking at how sound waves, the higher the frequency, the closer the waves are together. The waves are closer together. Low frequencies, the waves are spaced further apart from one another. I put all these pictures in, but here you can see the general pathway. This is the sound wave. This is a non-liquid fluid. Air is non-liquid fluid. And go to a bike shop and tell them you want to get some non-fluid liquid in your tires. They look at you like they're crazy. Tell them like, oh, I want some non-liquid fluid for my circular sleeves. They look at me like I'm crazy. Just want air in my tires. But depending on the frequency, this is going to vibrate. Depending on the frequency over here, the amplitude over here is going to be transferred to the tympanic, to the malleus, the incus, the stapes over there, transfer the energy into the vestibular duct, and then the energy exits the tympanic duct. But then this is the paralymph here, the endolymph here, but depending on the energy can be transferred over. This structure over here is the structure that's actually producing the lymph fluid, is where that fluid is actually being produced, that point over there. Other organs of sensation is olfaction. That's for sense of smell. There's the olfactory organs. They're located in the nasal cavity on either side of the nasal septum. This region up over here, very close to the superior meatus. And each of these different cells are going to be responsive to a different chemical signature. If that chemicals in the air, they will get into your nasal passageway. They will dissolve into the water on the mucous membrane, and that once that happens, if that chemical activates one of those cells, that cell is going to create a action potential, and that action potential will be transferred to the olfactory bulb, and in the olfactory bulb, a specific nerve will take that chemical smell to the cortex, cerebral cortex, and the cerebral cortex will look through its dictionary of smells and associate that smell with a particular word. And then that information is sent to your cerebral cortex, and you will understand what that smell is, which that smell can activate your digestive system. It can also activate your repulsive system. Like maybe it's going to activate your fight or flight syndrome because you want to just get away from that smell because you walked into a restaurant that like really stinks and you just want to walk out right away. So olfaction can activate different responses. It can activate rest and digest. It can activate the fear response. It can activate the fright response. In terms of the olfactory organs, made up of 2 layers, the olfactory epithelium and the lamina propria. Lamina propria is just the loose areolar connective tissue that connects that layer to the internal layers up to the bone. Olfactory epithelium consists of the sensors. These are highly modified nerve cells, and each of them are going to be specific to detect a distinct chemical signature. That chemical signature will bind to a ligand-gated channel. Once that ligand-gated channel is bound, then that sensation is going to be generated as an electric signal and propagated in a nerve. There's also supporting cells that are present in the epithelial structure, and also there are stem cells that are present. So as these cells get worn out, these cells can be replaced as necessary. And then the lamina propria is going to consist of the areolar connective tissue, a conduit for the blood vessels and the nerves and lymphatics, and also the olfactory glands, which are secreting the mucus. Okay. So each of these nerves is going to go through the olfactory foramina of the ethmoid bone, go to the olfactory bulb, which is on either side of the crista galli of the ethmoid bone, and then go into the olfactory nerve, which is cranial nerve number 1, and get to the central nervous system. But most of this is going to take place up in this area here. Here's your nasal septum at the point over here. And it's going to take place up over here in the superior meatus is the major location of smell. So olfactory gland, bony structure over here. Here's your pruniform plate here. Olfactory foramina, this is the pathway for the nerves, the olfactory bulb. And then from the olfactory bulb, it's going to lead to the olfactory nerve. Or actually, I should say the olfactory nerves collect up in the olfactory bulb, and then we'll continue on to the brain. But these are the nerve cells, and they will have their dendrites that go to the surface. The air molecules, the chemical molecules in the air will solubilize, and that ligand will bind the receptor. If that ligand binds the receptor, then this nerve is going to send a signal up, and it's detecting a particular smell. This is detecting another smell. This is detecting another smell, and another smell, another smell. Like this might be your chocolate sensor. This is your coffee sensor. This is your vanilla sensor. This is your sweet sensor. All of these are important because they can activate your sympathetic or parasympathetic responses. Then the olfactory gland is just producing secretions of mucus that keeps this area over here always moist. One, so that the tissue does not dry out, because it's the external environment coming in. You're breathing in air. That's the external environment. And you want to keep that air moist so you don't damage the cell's structure. Olfactory reception begins with the binding of the odorant to a ligand-gated channel. That ligand-gated channel is going to activate a G protein coupled receptor, and that's going to create a depolarization. That's going to open up a sodium channel, and that it's going to create a depolarization event, and that is going to result in a nerve pathway. The olfactory pathways, you have the afferent fibers. They're going to leave the olfactory epithelium, collect into 20 or more bundles, penetrate into the olfactory foramina of the cribriform plate, and reach the olfactory bulbs of the cerebrum. And they're going to synapse from the olfactory nerves to the olfactory bulb, and then the olfactory bulb is going to lead to the olfactory nerve, which the olfactory bulb is the olfactory nerve, and that's going to bring the information to the brain. So the olfactory pathways, leaving the olfactory bulb, travels to the olfactory cortex, to the hypothalamus and the limbic system. All of these different systems are going to be alerted. The hypothalamus is going to be alerted because you want to know, is this going to be a flight or fright, or is this going to be a rest and digest type of smell? Then also the limbic system, is this a happy smell, such as the smell of dark chocolate and vanilla and sugar, or is it going to be a saddening smell? Such as you walk into the room where you've had a little puppy that's brand new and the puppy doesn't know any better, and you smell something and you go, gosh darn, I got to clean something up now, as soon as you find it. But that's what your limbic system does. That's your emotions. It's going to either make you happy, make you sad, or make you just not care at all. So olfactory information is the only type of sensory information to reach the cerebral cortex directly. Other sensations are going to be relayed from the thalamus. So it goes to the olfactory cortex, but also goes to the hypothalamus limbic. Visual information is going to the thalamus first. The thalamus is going to go to the visual cortex before you become aware of it. Olfactory discrimination, you can distinguish 1,000 different chemical stimuli. Dogs have 72 times more olfactory receptors than we have. We have about 200, about 2,000 different ones. They have way more. So their sense of smell is about 10,000 times better than ours. And elephant is about 5,000 times more sensitive than a dog. Elephants have the greatest sense of smell. But look at that giant nose they have also. In terms of the olfactory receptors, they replace frequency because they're under constant assault from the environment. But as you get older, the number of sensors does decline with age. Taste buds, just another type of sensory nerve cell. You have your oral cavity, you have your lingual epithelium, you have the muscle down below, and then emerging up through the field of the tongue are going to be the papilla, and that's really what these are. These papilla are going to be filled up with these different taste buds. And there'll be openings, and a chemical signature will come in, and that chemical signature will bind a specific type of protein channel. If it binds that protein channel will send an action potential on an afferent nerve and to the hypothalamus or to the thalamus. And at that point there, you become aware of that particular taste. We had the, circumvallate papilla towards the back of the tongue. There was the fungiform, and then the, what was the third type? I can't remember the third type. Filiform papilla. The filiform are the major field of the tongue. But in terms of like different areas of the tongue, I mean, all areas of tongue can detect all different tastes, but the areas that detect the different tastes the strongest is the back of the tongue is going to be detecting more of the bitter tastes. You can detect some bitter tastes here, but mostly the bitter tastes are going to be detected over here. More of the sour taste, these reached over here, the salty at that point over there, the sweet over here. But you do have at the tip of your tongue, you do have overlapping for sweet and salty occurring. So bitter, salty, sweet, sweet and salty. Then you have your vallate papiform, or circumvallate papilla, the fungiform papilla, and then also the filiform papilla, which are not depicted on there. Okay, well, that's it for today. Got all like 60 chapters done. Save, yes. So anybody have any questions? Well, if not, if you have questions during the week, send me an email. If you want to know how you're doing, what you need to make on the final, send me an email and I can give you a very good idea. I don't have your lab exam 5 entered in yet because it's not due until the 3rd. So I know some of you done it. I don't know if you plan on doing it again. I know some people made a 90 or 94. I don't know if they want to do it again to make their grade higher. I don't know. So I'm not going to enter those grades in until probably Saturday. I get those grades entered, but I can give you a fairly good estimate of how you're doing and what you would make on as a final grade if you got a 70 on the final. And then I could let you know like what you need to go up a letter grade. So if you want that information, let me know.
Do we have class on Monday?
Nope. Just the exam on Wednesday.
So we meet at 9 a.m. on Wednesday?
Yes, 9 o'clock. 9 a.m. is the scheduled exam time. It's based on room availability, so is where they base the schedule, but we're not meeting in rooms, we're meeting in rooms at home or rooms at Starbucks, wherever you might happen to be. I don't know where you are. So until Wednesday of next week, or emails or text, whatever, I will be here at your service.
Okay, thank you, Professor. Have a good week.
Thank you.
Thank
Have
you.
a nice week.